Okay, but like, I think mine picks up external values.
This phylogeny, the basic phylogeny of the eukaryota. And so we've looked at this phylogeny. Now, I haven't put the individual groups up here for Staminophyla that we went over, but there's 3 that we covered. And Alveolata, there's also 3. And then we're going to just briefly talk about the Archaeplasta, Amorphia, and then I'm going to the next lecture, which will concentrate on the Archaeplasta. And so these are where we're going. And just to review where I think I left off, we covered of the chrome alveolata. We covered 1, the Staminophyla. And the apomorphy for the Staminophyla is that they have 2 long or 2 flagella, 1 longer and tinseled. Okay, and as I said, what the tinseling is there's cytoplasmic strands that are coming, tubes that are coming off of one of the flagella, and this is characteristic of all Staminophyla. And then we looked at the various groups there. We just got into the Alveolata. The Alveolata is believed to be the sister group of Staminophyla, and the main apomorphy that these have, besides having a fair amount of DNA evidence to support the monophyly of that group, there are these membranous sacs that are located just right under the plasma membrane, and these are called alveoli. All right, but and so we went over the dinoflagellata. Okay, and even in lab you've got to look at some of these, and so I won't concentrate on them. I believe that this was the last slide that I covered. Is that correct?
Yes.
You covered.
Get to 24. Anybody? Yeah, no. Okay, just till 23. Okay, that's what I thought. All right, so we'll continue now from here. Okay, dinoflagellates. Incidentally, these and diatoms, these 2 groups are one of the major photosynthetic organisms in marine habitats, further away from the intertidal zone. In the intertidal zone, the main photosynthetic organisms there happen to be the brown algae and the red algae, which I'll get into a little bit later here. All right, dinoflagellates. Okay, apicomplexa. These are only found as parasites in animals, and there's a wide variety of them. Some of them cause, for example, some cats will have a disease called toxoplasmosis, which sometimes can be transferred over to humans, and that's due to an apicomplexan parasite. But the type of parasite that is most well known with the apicomplex are those that cause malaria. And there are species that will cause malaria in birds. There are 4 different species of Plasmodium, which is a genus that belongs to the apicomplexa, that cause human malaria. And so forth. Now, these have a real complex life cycle. And I'm going to later on go over the life cycle, typical life cycle of one of these malaria parasites. But there's also another structure that they have, namely the reasons they're called apicomplexa, because they have this very complex apical structure, what they use to penetrate and get into the cells that they invade. Now, the figure I have up here, you also saw this in lab, red blood cells, human red blood cells. And let me get my pen. Human red blood cells. And this is a lot of the slides that you had in lab that I looked at were pretty poor, but this one here is a pretty good figure. And what the parasite is, in fact, right here, this is called the ring stage, and it's merocytes. I'll talk about them a little bit later on, but it's not just one. There's a bunch of them that are forming a circle here. So these things will penetrate, they have this apical structure, they penetrate into the cells, and they live inside of the cells where they're eating and digesting proteins and reproducing asexually inside of those cells. And so now, one of the things about them, of course, they belong to the chrome alveolata. So that means that they're in their ancestry far back, they had ancestors that had red chloroplasts. Do we have any evidence of that for this group? Because they don't have chloroplasts, but they do have non-photosynthetic plastids. Definitely have that. And so I'm not marking that as an apomorphy, because that's really the possession of the plastid alone is a chrome alveolate character. Okay. Now, instead of this plastid being used for photosynthesis, it is for fatty acid metabolism. Therefore, it still has some functionality, but we have little question that it's actually derived from a chloroplast. Okay, so this group goes through many different forms. Okay, even within the same species, and early on, these different morphologies made it look like they were different species. But once we understood the life cycle, we could figure out that really these are different forms of the same species. Yes, Abani.
Are different functions considered apomorphies?
Is different? No, because fatty acid metabolism is also a characteristic of, well, that's mitochondria, not chloroplasts. I don't know if regular chloroplasts are involved in fatty acid metabolism elsewhere, but here is the only function that it has in this group. Okay. So I'm not counting it as an apomorphy, no.
Okay.
Okay, so one of the life stages that these have are called sporozoites. So the sporozoite happens to be haploid, and this is the form that gets inside of the cells. It's the one that has the apical structure. And so their life cycle, which we'll look at a little bit later in this lecture, in fact, one of the last few slides is involves both asexual reproduction, that is, they reproduce by mitosis, and also at one point sexual reproduction as well. So very complicated life cycle. So as I said, these are one genus here causes malaria. And in this case, it's the genus called Plasmodium, and I'll show that a little bit later on. You don't need to know that now. But within this, how does malaria get spread around from one person to another? It's by the bite of a female mosquito. Only female mosquitoes happen to imbibe blood. Male mosquitoes do not. It's only the females that are biting. Furthermore, it's not all mosquitoes. It's a narrow group of mosquitoes that belong to one particular genus, namely Anopheles. And even within Anopheles, I think there's probably well over 200 species of Anopheles mosquitoes. And it's only certain ones of them that are capable of transmitting this disease. And later, as I go over the malaria life cycle near the end of this particular slide episode, then we'll look at how that works. All right. Okay, another group that now belongs also to the alveolata, and this is Coleophora. In Coleophora, these are called, the common name is called ciliates. It's a relatively large group, and a lot of them are freshwater, some of them found in soil. And they instead of having flagella, their bodies are covered with cilia. And so this is not an apomorphy, because other groups of eukaryotes also have cilia as well. Okay, but this is the apomorphy for them. They have a really interesting life cycle, which I'm not going to go over, but nonetheless, they have two different types of functionally different nuclei. The two nuclei still have the same genome. Each of them has the same genes, however they function different from each other. One of them, called the macronucleus, during its vegetative growth and normal life, that is what it controls the metabolic processes in the cell. This is from this macronucleus is where the protein encoding comes from and everything like that. The other nucleus still has exactly the same genome, but it's called a micronucleus, and it's only involved in sexual reproduction, which we will not go into the life cycle of how these guys reproduce. It's another different and not so complicated life cycle, but it's complicated enough. But we don't have time to look at all the different life cycles that happen to be available among these organisms. All right, so paramecium, which you saw in the lab last week, these are typical, or you will see them. I'm sorry, they haven't come in yet. You will see them on Monday, but they're typical members of this group. Okay. All right, so that takes care of the chrome alveolata, the two groups. Now, what is the sister group in SAR? Okay, sister group in SAR to the chrome alveolata is something called rhizaria. And these, the apomorphic for them is that they're amoeboid. They're sort of, that's the body form they have, is like an amoeba. They have pseudopodia, but instead of the lobate pseudopodia, that is pseudopods that look very broad and wide, they have very narrow needle-like pseudopodia. And there's several groups that belong to the rhizaria. We're not going to talk about all of them. We're just going to concentrate on just one called the foraminifera. Okay, so keep it straight, even though I have just only one clade within the rhizaria. You need to look at the lineage here as being Eukarya, SAR, rhizaria, and foraminifera. And the apomorphies for rhizaria, which is a broader clade, includes other clades that we're not talking about, is not the same as foraminifera. Okay, indeed, foraminiferans have needle-like pseudopodia, but so do some other groups that belong to the rhizaria. What is unique to the foraminifera is that they secrete an outside of the plasma membrane, an organic shell. Okay, it should be called a cell wall, but no one seems to want to call it a cell wall, simply because these are heterotrophic. Usually it looks like cell walls, or use of that term is restricted to just only photosynthetic organisms. But anyway, this is still an organic material that is chalk, okay, calcium carbonate. Calcium carbonate associated with the organic material. And so this is multi-chambered cells. And you saw this in lab. They look like snail shells, but they're very, very small. And so now how can they do this? If they have a shell on the outside, calcium carbonate, and they're living inside of that shell, how do they get their food? And how do they stick their pseudopodia out? It's because there's tiny pores in that calcium carbonate, and the pseudopodia in the living animal is sticking out beyond the shell, and that's where they're taking up bacteria and other organic materials by phagocytosis, endocytosis, which then moves down the pseudopodia into the cell itself. This is a group that's exclusively marine. There are no freshwater members of it. And furthermore, about 90% of those species that we know of are from fossils. During the Cretaceous period in the Mesozoic, they were pretty abundant in the oceans, and their diversity really dropped with the end of the Mesozoic extinction. And we still have some today, but they're not very abundant. Certain oceans in the past had a lot of these growing in them. The deposit is chalk. So this type of rock, sedimentary rock, called chalk. And in fact, we mine it for actually chalks when we used to have chalkboards, right? But chalk is used for other things as well. But this is the remains of, or sediment that contains high amounts of fossilized foraminiferan shells. All right. Okay, so that's two of the so-called supergroups. And I will just, yes, Alquazim. And then Avanti.
Yeah, sorry. So, so they are the ones that are accountable for making shock formations, and not like the other way around.
That's right, because the chalk formations, they're up in the water column. Okay, they're not on ground. They're tiny. They're floating up in the water column, and when they die, their chalk shells, now that they can't propel themselves and keep them afloat, they will sink down and become part of the sediment. And so if you have a rich abundance of these organisms in the water column, they will set rain down, forming the sediment. And eventually that sediment, it's a sedimentary layer, it will also have calcium carbonate that fuses those shells together, and we get a type of sedimentary rock that's called chalk.
Wait, isn't limestone also calcium carbonate?
It is, but it's mostly calcium carbonate, but not in the doesn't have the organic material that makes the chalk a little bit different.
Okay, thank you.
Yeah, because they're chemically different. They are chemically different. They both indeed involve calcium carbonate, but the other components that are involved in there, calcium carbonate in this case is not the cement between sediment particles as it is in limestone. Here, the calcium carbonate is primarily in the chalk shells themselves, as well as the cementing material that holds those chalk cells together, chalk shells together.
So is that cementing material also made from them, or is that separate?
Say what?
Is the cementing material also made from like previous generations?
So we're talking about two different compositions of calcium carbonate. Once they incorporate calcium carbonate into their shells as they are living, okay. But once they have died, and what cements the calcium carbonate that cements them together is just calcium carbonate that's dissolved in water that precipitates out.
Okay, thank you so much.
Okay, Abani.
Could you explain what pseudopodia are?
Pseudopodia are finger-like extensions of the plasma membrane that a lot of amoeboid organisms use to take up their food by endocytosis.
Okay, and are forum like another clade under foraminifera?
Well, foraminifera is the official name for forams. Forams is what you call these critters as an adjective.
Oh, okay.
Okay, same thing. So someone here is asking in chat, is malaria the same as dengue? No. Malaria is caused by a eukaryotic parasite, Plasmodium, and a few other species. Dengue is it's a virus. It's a viral disease. It's also spread by mosquitoes, but not necessarily Anopheles mosquitoes. It is spread by other types of mosquitoes, primary Aedes aegypti, which is it belongs to a totally different genus. Aedes aegypti is also spreads yellow fever. It's the vector for yellow fever as well as for dengue. Dengue is a virus that belongs to the same viral group as yellow fever. Okay, but and it also not only Aedes aegypti. I think it's also Aedes. What is that critter? I don't remember. Don't remember the species name, but nonetheless, they're not the same disease. Okay. All right. So now, supergroup Archaeoplasma. So here I'm just going to mention a little bit about them, and then in the next lecture we're going to zero in on this group. Okay. So this is the group that has a primary plastid chloroplast that originated from primary endosymbiosis, where the chloroplast is very, very much like a cyanobacterium, and it preserves quite a bit of that. Early in the evolution of this group, so we had some sort of heterotrophic eukaryote long ago. In fact, this group, the fossils go back at least close to 1.7 billion years. We have archaeoplastids. Mostly, no, they were, I think the earliest ones might be green. No, I think they're red algae, but nonetheless. Soon after, of course, they had, when we say primary plastid here, they have chlorophyll, originally chlorophyll A. In fact, they all have chlorophyll A. And it's two lineages then split, which one we call the green lineage, the other the red lineage. The red lineage has chlorophyll C, which is unique, okay, because the green lineage does not have that alternate form of chlorophyll. The green lineage has chlorophyll B. And so which one of these came first out of either one of them probably evolved from chlorophyll A, but we don't know exactly which. Okay, so here the main apomorphy, synapomorphy, is that they have primary endosymbiotic chloroplasts. Okay, now interestingly, what belongs to the green lineage, I believe it belongs to the green lineage, but it's a group that I've already mentioned, glaucophytes. I'm not going to put them in here, but they have in between on the chloroplast, they have a thin layer of peptidoglycan between the inner and outer membrane in their chloroplast. which is another good indication that they evolved, that chloroplasts are derived from cyanobacteria. And I'll have another one here in a moment. So the major clades here in Archaeoplasta, Rhodophyta, these are called the red algae, and Chlorophyta, green algae. I'm still going to keep this as a clade, but I want to mention it's paraphyletic. This is a group that is close. Some chlorophytes are closer to land plants than others happen to be. And if we take out the land plants, then it makes Chlorophyta paraphyletic. This is in the process of being divided up, but it's still a lot more studies that have to really figure out the correct relationships among these chlorophytes. Okay, and then of course planting, the land plants. This is and when we talk about land plants, they're terrestrial. Okay, they grow on land. There are water plants that have returned to water, but we don't find land plants growing in the ocean. This is totally fresh water and also on land. Okay, and that's where we're going a little bit later here. We'll leave that alone. And the other supergroup, Amorphia, it's also recently had been called Uniconta. In fact, it's been called Uniconta for a long time. It's but this is now the name by which it goes, but you'll still come across this word Uniconta. You may even come across it in my slides here where I have not corrected it yet because this is a relatively new name. And once again, this is going to be another supergroup that we're devoting several other lectures to later on. The main character here that holds us together is a molecular character, but instead of saying that it's the gene sequence or the nucleotide sequence, and you have this 100 base pair piece that you have to memorize as an apomorphic for this group, this here is clear. Okay, so this is called the triple pyrimidine biosynthesis fusion gene. Now that might seem that's a lot to have to say, but what is a pyrimidine? Does anyone here remember what pyrimidines are from last semester?
Gene C, right?
I think it's single, right?
Yeah.
Single.
Is it like a single ring instead of two?
Nitrogen base, single ring. So uracil, cytosine, and thymine. These happen to be pyrimidines, but they are made from other materials. And so they're the end product of a biosynthetic pathway, where each step of the pathway requires a different enzyme. One of these enzymes is carbonyl phosphate synthase. Another of those enzymes is called DHO. That's an abbreviation. We don't need the acronym. We don't need to go into what it means. Another one is ACT. And these genes that code for these enzymes are normally separate from each other. Okay, in all other eukaryotes, they're separate genes. But here in Amorphia, they have fused together. We get one transcript that is then translated in the protein and somehow gets cut up into these three different enzymes. Okay, so this is what we mean by fusion gene. And this is the only one that we have here in Amorphia. Another apomorphy is that they all possess one flagellum. Okay, earlier I said probably the plesiomorphic case, plesiomorphic state for the number of flagellum in the eukaryote. It's probably two. There's very strong evidence for that, even though in some excavates there's more than two, but still two seems to be pretty common. Here, however, it's been reduced down to one. Okay, and so that loss of one flagellum, I'm counting that as an apomorphy. Okay, primitively heterotrophic. Okay, ancestrally heterotrophic. There is no evidence of any members of the Amorphia ever having ancestors that were photosynthetic. There's no genes, there's no plastids anywhere. And so they were exclusively heterotrophs. And there's several clades, but we'll be looking, when we get into Amorphia, looking at three major groups. One is called the amoebozoans. These are called that include lobate amoebae. Okay, that's different than the amoeba that we find in Rhizaria. Okay, in Rhizaria, those have needle-like pseudopodia, but these are rather, in the lobate amoebae, rather broad finger-like pseudopodium. And then something called slime molds. And I'll talk about them when we get there. We don't need to dwell on it now. And the fungi. Okay, this is a very major group, very important group. And so I'll have one whole lecture on that will include amoebozoans and fungi together, and then several that will cover animalia, okay, the animals. And fungi and animals are very closely related in their evolutionary history. Okay, so that gives an outline of all the major branches, and then of eukarya. And later we're going to concentrate and go into more details on just two of these branches, namely the Hemorrhaea and the Archaeoplasta. But before I leave these algae and other groups of eukaryotes, I want to just go over some eukaryotic life cycles. They're very, very varied. And one of the things you get with our textbooks when we go through Biology 101A or in similar types of biology classes is you don't get a real strong feel for all the diversity and life cycles that happen to be out there, because we mostly concentrate on animals per se. And just to review that, okay, I'll put up here some review what the animal life cycle is too. So although we covered mitosis last semester, it was basically what happens in animals and also in plants. But among some of these other groups, they have some unique forms of mitosis where, for example, some groups, during mitosis, the nuclear envelope does not break down, and yet the spindle apparatus forms outside of the nuclear membrane. Asexual reproduction among a lot of the eukaryotes that we've talked about is very widespread. Sexual reproduction also. However, a lot of times what happens with sexual reproduction is it produces a resistant form that can withstand the environment drying out. And when the environment returns back to a habitable situation, then meiosis will take place, and the so-called cyst will result in zoospores or some other form growing back to their normal life cycle. Okay, several different life cycles. I'm only going to go over 3 of them, but just as a review, just to remind you what an animal life cycle is like. It's a diploid life cycle, and so all of the cells in the multicellular organism is diploid, and the only haploid cells happen to be gametes. So it's a diploid cell, a germ cell that undergoes meiosis, like we covered last semester, producing a haploid sperm cell, a haploid ovum. Ovum is a better word for egg, okay? And with humans, of course, these are haploid 23 chromosomes. This is the only haploid stage in animals. Then fertilization takes place. It's also called syngamy. That's going to be a word that will come up on some of the later slides here. Same thing. The fusion together of the sperm cells and the ovum, they join their chromosomes together, and we have here now the diploid zygote, which then undergoes mitotic development to grow into the adult. That's typical life stage. Now, diatoms, they do have a diploid life cycle. However, these are unicellular organisms, and their life cycle is a little bit different than what I'm talking about for animals. Once again, the lineage. And I like asking lineage questions. It'd be Eukarya, SAR, Chrome alveolata, Straminophylla, and Bacillariophyta. Okay, that's lineage. And so they're diploid, of course, and one of the morphological things I talked about is an apomorphy, is that their cell walls contain silica, okay? It makes them glassy. Furthermore, that cell wall comes in 2 halves, where one half fits over the edges of the other one, sort of like a box in a lid. And that's an apomorphy, okay? Now, when they undergo mitosis and they divide, each half of this cell wall, the box and the lid, they will be inherited by the 2 offspring. So one of the daughter cells will inherit the lid, the other one inherits the box, and then it has to secrete a new other half. And the new secretion is always a box, okay? Now, as a consequence of this type of cell division, after several mitotic divisions, some cells are becoming smaller and smaller because always, it doesn't matter. We have a box, we have a lid. When it separates in the daughter cells, both of them, including the box, which is smaller than the lid, the box will become the lid of that particular cell, and it secretes a new box. And so this cell is smaller. And if that keeps going, they keep getting smaller and smaller. And then that's when they'll undergo meiosis, is when they get too small, because meiosis will restore their original size. And so if we go to this next slide and look at it, this is how it works. So there's it's a diploid, okay? 2 sets of chromosomes, and I'm not showing the nucleus in here, but here is the lid, there is the box. And as it undergoes mitosis, the green is the box, and that is what is newly synthesized. Each of them inherit the box and the lid of the parent cell, and that will always become the lid. And therefore, this individual here is a little bit smaller than this one. This one here is the same size as the parent from which it came. All right, now if we go ahead and go to the next, it goes through another mitotic division, and you see this one right here, okay, is getting smaller. That is smaller, that is smaller. And if we keep going, here I'm not going to worry about the upper two, just look at this real small one. We still are getting yet smaller and smaller. Okay, so once it gets small enough, then it will undergo meiosis. Okay, it's a single cell. And so here the whole cell functions as a germ cell, and therefore undergoes meiosis, sheds the silica cell walls, they become part of the sediment. Okay, now this is, you notice that these cells don't have flagella. It is the gametes, the sperm, that have flagella. And this is how we know that they're straminophiles, because they have, I didn't put it here in my drawing, but they still have 2 flagella. One of them, the longer one, is tinsel. And I didn't put the tinseling in here because it was just too much time to draw it. Okay, so now these can be male and female cells. Female cells, when they undergo meiosis, produce an egg which does not have flagella. It's only the sperm cells that have flagella. So up here, this was a male, and this here came from meiosis in a female. Okay, we can correctly call them males and females because the egg and sperm are quite distinctive. And so when they fuse together, it produces, so once again, the sperm cell and the egg cell is the only haploid stage in this group, produces a zygote, which is now diploid, and that zygote will restore back to the original size. It'll secrete new cell walls eventually. So this is diatoms. Okay, now go over also haploid life cycles. And this is typical of Chlamydomonas. Chlamydomonas belongs to Chlorophyta. Okay, it's a green alga, and it's a unicellular green alga. And most of the time, so their vegetative normal condition is to be haploid, 1 set of chromosomes, just 1 set of chromosomes. And they will undergo mitosis to asexual reproduction to produce other cells that are, these are biflagellated, incidentally. They have 2 flagella. Now, when it undergoes sexual reproduction, each vegetative cell will then function as a gamete, and they fuse together. But they have to be of different genetic strains. We characterize that as plus and minus simply because they look alike. Okay, we can't tell which one is egg, which one is sperm. They both look the same. They both have flagella, and when they're functioning as gametes, they look just like the normal vegetative cells that are undergoing mitosis. But we do know that they have to be genetically different. We never have a mating here between 2 plus strains or between 2 minus strains. It doesn't work. They're genetically excluded from having reproduction like that. So here they will fuse together, and this is called fertilization. Is also called syngamy. It's a word we'll come across, and it forms a diploid zygote. Okay, this diploid zygote usually is encysted. Okay, it has a very hard shell on the outside, and here the puddle or pond in which these organisms are growing, because they're freshwater, can dry up, and these are very resistant. They can stay quiescent until the water returns, and when they do so, they will undergo meiosis to produce then the normal vegetative cells that look just like, and they do increase in size. They're small, but they will eventually grow. And out of this, because of the reproductive system, just like males, XY and XX chromosomes, although this is due to a gene, it's not due to a particular sex chromosome, that you will produce two out of this, produces four, meiosis results in 4 cells, 2 of which are positive strain, 2 of which are negative strain. And then these will grow back to the normal vegetative type, and they will continue to divide by mitosis. So this is the normal life cycle. Okay? And I didn't put those words up there, so I better put them all together, and I'll give you some time to get them in case I took them off your set of slides.
Professor, so the vegetative state is what the chlamydomonas would be considered as the haploid, and then when a positive and a negative strand combine, they form the zygote that goes meiosis to reform chlamydomonas, right?
That's right.
Okay.
Exactly. And so they'll go through in a season, they'll go through a lot of cycles of asexual reproduction long before they ever go through any sexual reproduction. In this genus, Chlamydomonas is a genus, and several different species in it, that usually the zygote in sexual reproduction, forming a zygote, is usually when harsh conditions set in, when either the pond or puddle is drying out or it's becoming too cold. And they will overwinter this way.
And the asexual reproduction just by mitosis, and the positive strand will just produce more positive strand ones, or?
That's correct.
Okay.
Because they're haploid. They're haploid, and so positive strand, when it divides, will not positive strand, positive strain. Okay, strand is something different. The positive strain will produce only positive individuals, and negative will produce only negative individuals. That's correct. Okay, everyone have this. Okay, then, yes.
I have a question.
Sure.
So when you said vegetative state, it means the mature cell of the.
Yeah, and it's like you call it vegetative just because that's when it's doing its thing. It's living. Okay, it's not reproducing except for by mitosis, and so it's photosynthetic. It's where it's undergoing its normal metabolism. It's growing to a certain size. Then it'll undergo another phase of mitosis, but most of the time it's in that vegetative state. So I'm using this word vegetative state not as in the same way that we do with people in comas. You say it's vegetative. That's not what I'm meaning here. Okay, does that make sense?
Yep, thank you.
Okay, all right. So just a moment here. All right, now the last life cycle. This is of an apicomplexan, and it is the malaria parasite. So once again, I like to give the whole, if we put in the whole lineage in front of Chrome alveolata, you would have SAR, and before that you'd have eukaryote, and they would have the whole thing here. So these are all parasites, and of course the infectious part of the life cycle is a form called sporozoites. It's a sporozoite that has that apical structure for penetrating into the cells in which they invade. And so the life cycle includes both sexual and asexual phases, and I'm illustrating this. And also, it isn't the case that these apicomplexans, different genera have exactly the same complex life cycle. They all have complex life cycles. Some of them may have more than 2 hosts. In the case of Plasmodium, however, this particular genus that causes malaria in humans, it only has 2 hosts, namely the mosquito the Anopheles mosquito, and the human. But in some other groups of apicomplexans, they may have 3 different hosts that they live in, and have still very complex life cycle that involves 2 or more hosts. So the vector for this, there's that word again, vector, means it doesn't cause disease. The bite of the mosquito does not cause malaria. You get it's the way in which the malaria parasite is transferred from one individual to another is by the bite of Anopheles mosquitoes. And these are always female. The males, they're down at some flower sipping nectar all the time. They're not bloodsuckers. All right. So in humans, there's 4 different species of Plasmodium. One called Plasmodium falciparum. It gives the worst type of malaria. Yes, Ivana.
What did vector mean?
Vector is any agent any living agent that transfers what is called the disease organism, the etiological agent. Let's see here, just a moment. Okay, you have a vector, and okay, the etiological agent, this is the organism that actually causes the disease. The vector doesn't cause the disease. The vector is only an organism which is involved in the transfer of the etiological agent from one individual to another. It's the one that spreads the disease, but it isn't the cause. Okay, does that make sense? Okay, just like with African sleeping sickness, something I talked about before, it is the tsetse fly is the vector, whereas the trypanosome is the etiological agent. And so likewise here, mosquitoes are not born with malaria. Okay, when they hatch, they're clean. They don't have the malaria parasite. The only way in which they pick up the malaria parasite is to feed on blood of someone who has malaria. Now, one of the forms of the Plasmodium is something called a gametocyte. Now, the gametocytes look different than squirrel cytes. Okay, and so the gametocytes, they are functioning as gametes, as the name implies. Yes, Alcocin.
I know you mentioned that they are hosts to, that the hosts for this are both mosquitoes and the humans, but is it parasitic towards mosquitoes as well, as in like it's like negatively affecting them?
It does negatively affect them. Sometimes it's hard to tell because you can't come up to a mosquito and ask them how you're feeling today, like you can with a human. But nonetheless, they do produce these blisters on the inside of their intestine, in the cell side of the intestine, which I'll show here in a moment. And a lot of times the mosquitoes will become very slow if they have too many of those blisters. And so it does look like they are affected. And just being a host doesn't necessarily mean that they suffer. And so being a host of something, you just have the organism living in you, and it may or may not give you any sort of disease symptoms. You're still a host. Your host which humans can have. We'll get into certain parasitic animals such as nematodes, and sometimes those nematodes you never even know you have them. Unless they get real abundant, then they can cause symptoms. But you're still a host if the animal's living or organism is living inside of you. Okay, we're the host to all sorts of bacteria in our large intestines. We have many different species, and we're still the host. Even if a lot of these bacteria are normal for us. And in fact, many of these bacteria growing in our intestines is actually beneficial to us because they protect us from being affected by harmful bacteria most of the time. And a lot of times when we get a disease like Clostridium that's a bacterium that produces a toxin that gives you real bad diarrhea. It's because something went wrong with our normal bacteria that happened to be there and allowed space for the harmful bacteria to grow. So in that case, we're still the host. Okay, so female mosquito bites and takes up blood from a malaria patient who happens to have gametocytes circulating in the blood. Okay, so these aren't inside of any cells; they're in the plasma of the blood. And usually, a lot of times, these Anopheles mosquitoes, when they feed, it's at dusk. Okay, in the evening, not in the middle of the day. It's at dusk. And what is interesting here is that these species of Plasmodium have been selected for releasing gametocytes at dusk. Okay, because otherwise the immune cells would be able to clean them out. Okay, one of the right reasons why the Plasmodium parasite, the malaria, is hard to get rid of is because so many of life stages are inside of red blood cells where the immune system cannot recognize them. But these gametocytes would be recognized. However, they're there in high numbers, only very, very briefly. Okay, so if a mosquito happens to take them in, okay, you got that. I don't know why it does that. Okay, so it takes in at least 2 of these gametocytes. And of course, these gametocytes end up in the midgut. Okay, insects have a foregut and a midgut. Okay, the midgut is where hydrolytic digestion takes place. And that gut is only, or the cell surface of that is just only 1 cell layer thick. And so these 2 gametocytes will fuse to become the zygote. Yes, Alcosin.
So I've got a question. So when they're ingesting the gametocytes, do they have to ingest it from a human? So does it have to like crisscross when mosquitoes get it?
Yeah, it has to be human. If they feed on birds, there's different species of Plasmodium there, and those Plasmodia are not compatible. They might be compatible with the mosquito, but they're not compatible with living in a human. And so to take up, they would have to take up the gametocytes of one of the 4 species of Plasmodium that can infect humans. Other forms, like from chimpanzees, have their own unique. In fact, one of these, I believe, is vivax, Plasmodium vivax, is found both in humans and in chimpanzees, and it probably jumped from chimpanzees, was transferred from chimpanzees into humans. But there's other species of Plasmodium that are in chimpanzees that will not survive in human. And furthermore, if this was, say, a Culex mosquito or Culexides mosquito or even Egypti, Aedes mosquito, of those different species, and they picked up these gametocytes in their midgut just get digested. They cannot survive. Mosquitoes are not the same. Okay, and so from one mosquito to the next, their digestive systems have their own properties, and so it's only with certain Anopheles mosquitoes that these malaria parasites can get through. Okay, make sense? Okay, so we form a zygote, and I'm leaving out another stage here that goes through here. But this is the gut lumen, and it has epithelial cells there that are 1 cell layer thick. And then out here you have hemolymph. And so these zygotes, or a form that comes from the zygotes, usually it's the zygote itself, will crawl between the cells, form a blister on the hemolymph side, that is on the body side of the gut, and undergo meiosis. Okay, so this will undergo meiosis, and then many rounds of mitosis afterwards. Okay, the meiosis will produce sporozoites, haploid, but these will still divide to increase their number. Okay, increase their number by mitosis, and eventually this blister will rupture, and sporozoites will then swim throughout the body. In fact, this is what they look like. That's sporozoite, and end up anywhere in the muscles, in the reproductive organs, etc. There's no direction that they go, but sometimes, or many of them, will end up in salivary glands. Salivary glands are 2 large glands in the neck of the mosquito. And they're very, very important because the proboscis of the mosquito has 6 stylets that are very sharp. And also those 6 stylets form a feeding tube. And so they will drill into the vein or something near the surface. And they need to suck blood through a very, very narrow feeding tube. And to prevent coagulation, they inject saliva into the wound. And so these sporozoites are in the saliva that they inject in. And so these are like hypodermic needles that are walking around with this malaria parasite. It takes about almost a week, 7 days after the mosquito has ingested the gametocytes before the mosquito is infected. All right, because it takes this life cycle of the sporozoites that going through the producing the zygote and producing the sporozoites, getting into saliva glands takes some time. So if this mosquito happens to feed within, maybe 2 days after ingesting the gametocytes, it will not cause and spread the malaria. It's only after these sporozoites made it into salivary glands. So this gets injected, so it bites another human, and it injects these sporozoites in through the tube, that's proboscis. And then these sporozoites will go through the blood system, and the first place they will get out is in the liver. And so they will penetrate into liver cells, and this will form a new form. They look very different, merozoites. So sporozoites, merozoites, they look different and they function differently, yet they're haploid, and they've been divided, they come from mitosis, right? So the sporozoites in liver, once they penetrate, they divide by mitosis to produce the merozoites. Now, why do you think these things look different if they have exactly the same genes? Okay,
Do they have to have a conformational change to be able to affect the hemoglobin? Or is it
not a conformational change, but a change in gene expression. Okay, because once again, in any organism, especially eukaryotic organisms, they don't have all genes turned on. Okay, and so if you have a certain set of genes that are turned on, it creates a certain form, the sporozoites, and when those genes are shut down and other genes are turned on, then it takes on the form of a merozoite. And so it's a matter of gene expression, okay, and it's highly regulated. Okay, now these merozoites, once they break out of liver cells, they will then invade red blood cells, where they produce, once again, asexually, okay, by mitosis. And they penetrate into the red blood cells. And they're sort of hidden and camouflaged from the immune system because they can't be seen. They're on the inside, and they're eating up the hemoglobin and undergoing asexual reproduction. And that's what you see inside the red blood cells. These are stained. You wouldn't see them without stain, but they usually form a ring in which there's several merozoites. Now, when these merozoites, they're incidentally, their life cycles are synchronized. If this infection, go back here, was from the bite of one mosquito, so the red blood cells are analogous to the blisters in the mosquitoes? No. The blisters is a sort of a skin-like thing, bubble, and these are living inside of it until they divide, that the blister will break open. These red blood cells are, they sort of, because in that blister, they're feeding some, but it's on liquid hemolymph that's coming, that the blister is taking up. But here in the red blood cells, they have proteins that they're feeding on and other things inside. Okay, does that get your question? Okay, yeah. So these merozoites. Now, if this came from the bite of one mosquito, if these were all introduced by one bite of mosquito, we now have several of them in the liver, and they've gotten out of the liver. We've infected maybe hundreds of red blood cells. They are all synchronized so that they break out of the red blood cells, it lyses the red blood cells, it destroys the red blood cell all at the same time. And usually these cycles are 24-hour cycles or 36-hour cycles. It really depends on the species of Plasmodium, but they all lyse out at the same time. And it's during that time that the individual will suffer symptoms. They get a fever and chills, and that's because of all this red blood cell debris that happens to be around and the body's immune system is reacting to. Now, if an individual gets infected and bit several times by different mosquitoes, then he or she will be going through several different cycles of fever and chills, fevers and chills, depending on how many mosquitoes happen to get introduced. But anyway, here are merozoites, then invade other red blood cells, reproduce asexually, and eventually this builds up in the blood. Sometimes, however, if it's near evening, then some of these, instead of producing merozoites, it will, a burst red blood cell will still release both merozoites and gametocytes. These gametocytes will be in the peripheral blood system so that if a mosquito then feeds on it, then the gametocytes are transferred into the gut of the mosquito, and we've completed the cycle. So this is the life cycle of Plasmodium. Okay? And that is the end of this particular lecture. Okay, any slides that you need to see before I go to the next one?
Yeah, can I see slide 31? Also wanted to ask the lineage of eukaryote SAR that you mentioned. What was it again?
For
the one you mentioned on this slide. I think it was like eukaryote SAR something Stramina.
Yeah. In fact, the whole lineage here. Eukaryote, SAR, Biolata, and so forth. These are the two parts of the lineage prior to Chromalbiolata. And then the rest of it, of course, is then Stramnophylla, Bacillariophyta. Okay?
Could I see slide 25, please?
Slide 25.
Okay, thank you.
Okay. Is that it?
Professor, I have a question.
What's that?
I have a question.
Sure.
So for the last thing that we talked about, is the life cycle of like the one that we talked about the same thing as the trypanosoma, the say fly?
In the no, it's a different life cycle. Different trypanosome. I think you're saying trypanosome. That's also
that one.
parasite, but that belongs to excavata. You know, that's a totally different group, and they're not living inside of the blood cells. They're on the outside. Okay, they're in your blood plasma, and they're still eating blood cells and destroying them. Now, how do they evade the immune system? They have a dirty trick in that every so often they completely change their surface proteins, because that's usually what your immune system is making antibodies to, is that they'll see the proteins as foreign, make an antibody to it, the antibodies cling to it, and this immobilizes the parasite. But to escape the immune system, maybe every, I don't know, maybe 5-6 days, it completely changes the surface proteins it has in its plasma membrane, so that it's continually fooling the immune system. But yeah, they're totally different creatures.
Okay, thank you.
Yeah. Okay, so now we are setting here, get it up here. We'll then switch gears to lecture topic 5, which is then a survey of the archaeplasma. Okay, and here I'll be, as we go, especially when we get into land plants, I'll be talking a lot about their life cycles because they're very interesting life cycle of the plants. Okay, so the archaeplastid, this is green algae, red algae, and land plants. Okay, and as I already mentioned, the main apomorphy for the synapomorphy that holds these groups together is that they have chloroplasts of primary symbiotic origin. And so their ancestors took up a chlorophyte, or not a chlorophyte, excuse me, a cyanobacterium. And the cyanobacterium eventually lost its identity and became the chloroplast. All right, and so yes, Hamza.
Professor, did you upload the lecture notes onto Canvas?
No, he did not.
Yes, they should be there.
For 5, right?
Yeah.
I don't see them here.
No, maybe I did this because I frequently do this. I put it up and I have to have it published. So let me just double check. Right there.
It's probably something on my end. I'll just try
Yeah,
refreshing something.
because it's there.
Okay, thank you.
Oh, that's exercise. Wait a minute. Let's go up here to lab material. Maybe that's what I did. No, see, it's there. Let me make sure it's published. Yeah, it's published. So it's there in the lecture materials. Still can't find it? So it might be something on your end then. Okay, so the archaeplastida, they have a primary endosymbiotic origin. There's only 2 membranes around the chloroplast. There's not a third or fourth one. And furthermore, so just only 2 phospholipid bilayers. Furthermore, there's a lot of genetic evidence. There's a lot of that tie the chloroplast, green chloroplast and red chloroplast to cyanobacteria. So this clade, of course, includes the rhodophyta, the red algae, the chlorophyta, the green algae, and the plantae. The plantae is also called embryophyta, embryophytes, but we're going to use the word plants. Okay, plantae. That's most important here, or easier to recognize. So for this group called rhodophyta, we have fossils going back about I believe 1.3 or 1.5 billion years. And so some very early multicellular organisms happen to be red algae in the fossil record. And they're mostly multicellular, and there are some that happen to be unicellular as well. And in the past, some of the fossils look like we have a lot of them that is unicellular, but nonetheless, so they're chloroplasts. They, as you would expect, since these are archaeplastids that have primary endosymbiotic origin of the chloroplast, is just 2 membranes, and those 2 membranes correspond to the plasma membrane. Okay, that's the inner membrane of a cyanobacteria, and the outer membrane is the lipopolysaccharide layer. Although the lipopolysaccharide is gone, it still has beta barrels, and those beta barrels, porins, called porins, you only find them in the outer membranes of gram-negative bacteria or in the outer membrane of mitochondria or chloroplasts. Furthermore, of course, their chloroplasts, the pigments they have, something called, of course, chlorophyll A and chlorophyll C. Okay, I will put up here, I didn't put it up here, but you should put it in here, that chlorophyll C, we're counting it as an apomorphy for the red algae, chlorophyll C. Now, something that's also on their chloroplast, on the membrane, is along with the photosystems, because it's in the inner membrane of a chloroplasts is where we find in fact, it's inside, we have thylakoids. And of course, the thylakoids, in addition to having the two photosystems in them, in red algae, in addition to chlorophyll A and chlorophyll B, they have these branching structures on the surface of the thylakoids that are called phycobilosomes. And these contain pigments. These pigments, there's phycobilins and also phycoerythrin. Okay, now this is not an apomorphy. This is a plesiomorphy, because this is exactly what you find in cyanobacteria. In cyanobacteria, they also have thylakoids. Okay, it's one of the rare groups. The thylakoids are still continuous with the plasma membrane, and so it makes it appear they're not separate from the plasma membrane, but they produce a lot of surface area inside for photosynthesis. And in addition to having the two photosystems, as typical of chloroplasts, they also have these phycobilosomes. And so this is a plesiomorphy. It's the only group of eukaryotes in which the chloroplasts still retain these pigments. And it's because of the phycoerythrin that we usually find red algae further away from shore and in deeper water. Okay? Because this here absorbs blue light, and blue light will penetrate deeper into the ocean. And because you're absorbing primarily in the blue part of the spectrum, what you see mostly reflected would be red, all right? And that's what gives them their color. Okay, the cell walls, they do have cell walls, so the cellulose material, and they have a type of starch that they store outside of the chloroplast. Okay, it's in the cytosol. It's called Floridian starch. It's similar to starch in green plants. And so since these are algae, the complete body is called thallus. Okay, now, so in the thallus, of course, consists of a holdfast, okay, blades, and gas floats that keeps it up in the water. And so the thallus is a general word that we use for any multicellular alga. Okay, it's not just only brown algae, Phaeophyta, which is unrelated to the red algae, but also multicellular green algae that I'll get into here shortly. It's also, the body is called thallus. All right. Now, interestingly, too, nowhere in any part of their life cycle do they have centrioles or flagella. They do not have any microtubular structures that have this 9+2 arrangement of 9 microtubular doublets forming a cylinder with 2 microtubules in the middle, which is widespread throughout the eukarya. And in fact, we believe that this here, I already gave that as an apomorphy, the possession of this 9+2 microtubular arrangement. I gave that as an apomorphy for the eukarya. But this isn't the only place we're going to find it being lost. But since we expect eukaryotes to have this, its absence is informative. Okay, we can say that it's lost. So even in their reproductive cells, because a lot of times in these groups, for example, in the diatoms that we just looked at, the only time you find the flagellum is in the reproductive cells, okay, in the sperm. But even here, the reproductive cells of red algae don't have flagella. Okay, so that's what I'm putting here as an apomorphy. Okay, as I already mentioned, the phycoerythrin happens to be in the phycobilosomes. They can be in deeper water. Okay, and then the brown algae or the green algae. Okay, and these are just some representative forms, figures from your textbook. Branching, some of these, and here's another one, sheet-like, this filamentous. Okay, single cells. This here are single cells with branching filaments. You can even see the cell divisions here. This is under a microscope. But the sheet-like red alga is much, much bigger. And some of these kelps, they're called kelp, they can be well over 40 meters long. Okay, they can have incredibly large bodies. Okay, so that's all I'm going to say about the
Professor, real quick. Can you repeat what the phycoerythrin does exactly?
Okay, it's just like chlorophyll. It absorbs light, It's a pigment that captures light energy and channels it into the electron transport chain by moving electrons. But the wavelengths of light that it absorbs well at is blue. And so blue light, as light goes into the ocean water, it's the shorter wavelengths that get absorbed first. And therefore, the higher energetic light, blue, it penetrates deeper. Okay. And so you don't have too much light down at the level where these red algae happen to be. And so, but they can pick up the blue light there quite well. And this is why they appear red.
Isn't blue a shorter wavelength though? And you said the shorter wavelengths are absorbed first?
But it's a longer wavelength that gets absorbed at the top.
Okay.
So the blue gets through. And so why do these things look red? It's because they're absorbing blue.
Yeah, I see. So that's what they
that's what they transmit, yes. Okay. Now, it isn't the case that they're always deep. Some of them can be real close to shore, but they can still be found off of the coast in deeper water than what you would normally find green algae or brown algae. Okay. And of course, diatoms and dinoflagellates, they're not anchored to the substrate, and therefore they're not dependent on the substrate. They're floating in the water column, and therefore they can be far out away from shore, but still in the upper column of water where sunlight happens to be. Okay, so the rest of the plants, or the rest of the archaeoplastids, is a clade that's called viridiplantae. Okay, viridi is from Latin meaning green. Okay, green plants. Although these are not plants, they're algae and land plants. Okay, so viridiplantae, these are green algae, the chlorophytes plus the plants, and therefore an apomorphy for these is that they have chlorophyll B, and that's what makes their chloroplast the green chloroplasts in contrast to the rhodophyta, which have chlorophyll C and phycoerythrin, which makes them reddish, all right? Red lineage. Furthermore, another apomorphy here is that they store excess carbohydrate in the form of starch, but in the chloroplast. So it isn't just that they store it as starch. Red algae have a form of starch called Floridian starch, and also the brown algae have a certain form of starch as well. But in all these cases, the starch is being stored outside of the chloroplast. And so this is the apomorphy. The starch is in the chloroplast. Okay? So viridiplantae. So if we're going down here, let's go back. Let's come back to the slide. Let's get rid of everything because we're doing the clade here. In our plants, we have rhodophyta, or not plants, but the archaeoplastids, rhodophyta, and then viridiplantae. All this up here is viridiplantae. I'm just going to abbreviate it because it takes too long to write it. And therefore, we have chlorophyll B as an apomorphy for viridiplantae, and also starch in chloroplast. Stored apomorphy. So, in a Linnaean system, Chlorophyta is treated as a phytum. And as I said, this here is paraphyletic. Okay, it's not a monophyletic group. We're still going to keep it as just understand that it's paraphyletic. And because of it being paraphyletic, I'm not going to give any apomorphies for it because it doesn't make any sense. So this is green algae, and they come in many different life forms. Certain ones of groups of chlorophytes, and I will put them, mention them a little bit later on, they are the closest to land plants, and they probably form the sister group to land plants. But not all chlorophytes are sister group to land plants. So these course characteristics, because they are viridiplantae, I'm still mentioning this again. They have the chloroplast of chlorophyll A, chlorophyll B, but really all viridiplantae would have it because it's an apomorphy. Food, a starch, and chloroplast, That's not an apomorphy of chlorophytes. It's a consequence of them being viridiplanty. Cellulosic cellulose, that I'm not mentioning that as an apomorphy either, because we find cellulose elsewhere or various similar forms to that type of structural polysaccharide. Chlorophytes probably evolved, and maybe even the rhodophytes, we don't know for sure. There's some suggestion that they may have evolved in fresh water, but definitely the chlorophytes probably did. And then what marine chlorophytes we have, they have returned to the ocean, and that seems to have happened many times. There's many different body forms in this group. Some of them are unicellular biflagellates. That is, they swim around in water due to their flagella, and they consist of one cell and have one large chloroplast. Some of them form circular colonies, okay, that where the individual cells happen to be a biflagellated cell, but they're all connected together as a sphere. Okay, I'll show you that here in a moment. And some of them are filamentous, okay, forming long chains of cells, sometimes branching. And some of them are multicellular. So this idea of multicellularity, you see, has been attained multiple times in the eukaryotes. Once in the brown algae, okay, again here in the chlorophytes, some of them, and in the red algae separately. This is all convergent because the red algae, although most red algae today are multicellular, their ancestors are probably unicellular, because we still have them. Okay, this is going to be something that you'll see Monday, okay, even though you'll look at pictures of it tomorrow as well. But as far as live specimens, this here is something called Volvox, and it is a spherical colony. It's made up of tiny little cells that are biflagellate. So one of those little dots here happens to be an individual cell, and these cells are shaped like this, and their flagella are pointing outward from the sphere, and there's cytoplasmic strands between that hold the cells together, and furthermore coordinate the beating of the flagella so that these guys can reorient themselves in the water column where they have the optimal amount of light, because they are photosynthetic. And so this is something called Volvox. The green things that you see inside of the sphere. So this sphere is one cell layer thick, and it's hollow on the inside. And so what's on the inside are daughter colonies, because sometimes these cells on the outside will break off, move into the sphere, continue to divide by mitosis to form the daughter colony. Eventually, the mother colony will die, and the cells will fall apart and release these daughter colonies. Okay, and this is multicellular. Okay, this is a multicellular green alga living in fresh water. It has things that look like leaves, things that look like stems, but those are not true leaves or stems. And we have a lot of them, like Chlamydomonas. I went over its life cycle, that haploid life cycle. Although you cannot quite see in this photo, well, this one right over here, you can see the flagella. All these have two flagella. And there's several different types of unicellular chlorophytes. Something that's little on a clade that within the chlorophytes that's a little bit more closely related to plants is something that you see right here. This is a close-up of something called Spirogyra. And you'll also see Spirogyra in lab on Monday. I've ordered live specimens of it. These are filamentous, okay, so they have repeating cells that are in a straight line. And they're called Spirogyra. The plant or the organism was named before the rock band was. Okay, so the rock band is named after these, but their chloroplasts are in a band that twirls around, spirals inside here. So this is actually chloroplast, that green part that you see. Okay, and you'll get to see those. Okay, so that's all I'm going to say about the chlorophytes. You should know them. And in fact, a lot of times when you see when it's been real wet, rainy, and you go out on the north side of a building, on the concrete, on the sidewalk, a lot of times you find a lot of little tiny green film there. A lot of times these are green algae. Okay, they can live in the places like that. The land plants. Okay, land plants are, this is going to be where we're going to spend most of our time in this particular lecture. They appear in the fossil record about 475 million years ago. The first plants, very, very small, and didn't have a whole lot of leaves or anything. The whole evolution of plants all through the Paleozoic and into the Mesozoic and into the Cenozoic has been increasing adaptation to life on land, to be able to survive on land. No problem with living in the water. Okay, in the water, there's plenty of water, but once you move on to land, you're in a desiccating atmosphere where even if it's has high humidity, it still will take water away. And furthermore, it has to get through the soil. And therefore, part of the plant has evolved a system of living in two different places, namely in the soil, where it would be getting minerals and water, and also in the air, where the carbon dioxide happens to be, instead of being bicarbonate in the ocean water or even in fresh water, because that's how carbon is obtained by many of these algae. It's not as a gas, but as bicarbonate dissolved in water. So increasing adaptation to land, and we'll see that. And it took millions and millions of years for angiosperms and gymnosperms, which are the best land adapted plants, to achieve what they've achieved. Okay, so these plants, of course, multicellular. And that multicellularity has evolved separately from, well, I probably can't say that. It probably not so, because there's a group of multicellular green algae called charophytes that I will show a little bit later here that were multicellular. So I'm not counting that as apomorphic, but nonetheless, all plants are. Okay, all plants have multicellular body, of course, eukaryotic, also photoautotrophic, and because they belong to the viridid plantae, they have both chlorophylls A and B. Notice that I'm not putting a double asterisk here. These are just consequences of being viridid plantae, all right? Just repeating these characteristics again. Cell walls, cellulose, and of course they reserve food in starch in chloroplasts. And this is unique. However, it's not unique just for plants. It actually involves one group of green algae. They have spores. Okay, plants will produce two types of reproductive structures, spores and gametes in their life cycle, as we'll get into. However, the spores have cell walls with a very hard polymer called sporopollenin. And there's only one group of algae, green algae, that also have spores with cell walls of sporopollenin. And so this is a unique apomorphy, but not for the plants. They're sporoclade, a little bit larger than the plants, as I'll show you a little bit later on. Okay, so adaptations to terrestrial life. They have to have a broad surface to collect the light as an energy source. And therefore, one of the problems here being in a desiccating environment, as the air happens to be, is they could lose a lot of water through this unless they have evolved something to prevent or slow down water loss. And so as an apomorphy for the terrestrial plants is that they produce, their epidermis secretes and produces a waxy cuticle. So this is a layer of wax that's on coats all the aerial parts of the plant, and it that is exposed to the atmosphere, cutting down on water loss. Now, we also have to have gas exchange, and so that's a little bit of a problem too. And so what the leaves or leaf-like structures have evolved is special cells called guard cells that form an opening called stomata. And this is for gas exchange. We can go ahead and put that as an apomorphy. Okay, I didn't put it here, but stomata for gas exchange. Furthermore, reproductive structures they have called gametangium. This is where sperm and eggs are produced. And I'll later on give you two different names because there's one type of gametangium that will produce sperm cells and another type of gametangium that will produce egg cells. And these are produced by mitosis, not meiosis. Okay, so but the thing here is when we have these types of reproductive structures, even in chlorophytes, they have gametangent too, but those are single cells. Here, it's a multicellular structure, and so a multicellular gametangium that's unique to the terrestrial plants, and furthermore, it's covered by a layer of sterile cells. All right, so what do we mean by sterile cells? These are cells that do not engage in meiosis. Okay, doesn't mean that they don't have bacteria in them or anything like that. It's just that they are not reproductive cells. They're only there to protect the gametangia. Yes, Aquasin.
So is gametangia kind of like a specific reproductive organ? Can you like say that kind of?
So it would be analogous to testes and ovaries in animals. Okay, analogous. Okay, not homologous. Analogous. It have similar types of function. Exactly. And we'll see that there's furthermore, after fertilization, the zygote, instead of being released or undergoing meiosis, it is retained in the parent plant and go into and it'll start dividing by mitosis and grows into a multicellular embryo. Okay, so that's the apomorphy, is that we produce embryos. This is one of the reasons that plants are called embryophytes or embryophyta. I mentioned that as well, because that is an apomorphy. They produce a multicellular young plant that is retained in the gametangium. So first of all, we have fertilization, produce a zygote. The zygote will divide, and this all happens inside of this multicellular gametangium. And it'll divide several times by mitosis to produce a multicellular embryo that is still retained in the gametangium. This will continue to grow a little bit later on, but it's the embryo in the gametangium that I have here as a apomorphy. Now, unfortunately, I'm not going to get to all the plants that we'll be looking at tomorrow. We're going to be looking at two different groups of plants, but nonetheless, I'll get this far, and then we'll catch up on Thursday, I hope, before we get into Monday. I mentioned last semester, I did go over plant life cycle very briefly, that they have an alteration of generations, okay, where there's two different adult plant forms that differ both in ploidy, both in what means of reproduction they use, and also in their what type of reproductive cells they produce. And they are morphologically different from each other. This is called that they're heteromorphic. Okay, we have two different forms here, the sporophyte and the gametophyte. Okay, let's just start here with the sporophyte, because what I said is we have fertilization, produces a zygote, diploid, and that will produce a baby sporophyte. It's diploid, and this is the sporophyte will the adult form. However, these do not have gametangia. They have a different type of reproductive structure called a sporangia. Okay, sporangia is plural. Sporangium, I-U-M, singular. This is a reproductive structure of sporophytes, and it's in the sporangia that germ cells, diploid germ cells, undergo meiosis to produce haploid spores. All right, these spores then are released, and if they find fertile ground where they can germinate, they will. When I say germinate, that means they divide by mitosis. They divide by mitosis to form adult form called the gametophyte. The gametophytes will have two different types of gametangia, which we'll get into later. I'm just going to leave it at gametangia right now. Gametangium is just one. Gametangia is plural. This is a reproductive organ, and it's in the gametangia, depending on whether this gametophyte is a male or a female or bisexual, by mitosis. It's left out here, mitosis. It will produce single cells, gametes, sperm, egg, which are genetically identical to the cells of the adult gametophyte because they're produced by mitosis. There's no change, there's no shuffling of the genes here. That happens up here with meiosis, okay? And so the gametes are genetically identical to the cells within the gametophyte. These gametes will then fuse we get diploid, zygote, and we complete the cycle. Okay, and I'll get into this probably before we get into the lab tomorrow. I'll just cover a little bit of it, and then on Thursday, I'll start here as well. So this is where I'm going to stop. Any questions?
Can I see slide 9, please?
Slide what?
9.
9. Okay. Okay, and I'm also canceling, or I'm going to have office hours not immediately after this, because I have to run to campus. So I'm going to have office hours between 2 and 3. And I'll send out a notification on that.
Can I see slide 3? Can I see slide 3, please?
Slide 3. Okay, so nothing else? You're free to go.
Slide 5.
Okay, which slide?
5, please.
5.
Can I see slide 2?
Slide 2. Okay, you done with 5?
Yeah, thank you.
Okay. 2.
All right, thank you, Professor. Have a good day.
Okay, all right. Okay, so I'll go ahead and close this, and we'll see you tomorrow.